Hey, thanks for tuning in to chapter 17. This is part 2 of chapter 17, Adaptive Immunity. We left off last time talking about antibody production, the roles that antibodies play in the humoral immune system. So I want to continue on with whatever other functions that antibodies can contribute to in the immune response. The antibody can bind to the antigen. And when it does, remember that the binding between antibody and antigen is extremely specific. So the binding of the antibody to the epitope on that antigen, very specific. Foreign cells and molecules are tagged for destruction. So the antibodies, which are depicted as these green Y-shaped proteins, are proteins that are produced by your B cells, and they go around and they look for and they bind to things that is determined to be foreign and that should not be there and therefore should be eliminated. So you can see that this antibody has bound to two different antigens at identical epitopes. And what this is doing is it causes an agglutination or clumping of the antigen. So in this case, the bacteria acts as the antigen. And one antibody, because it's got two identical antigen binding sites, can cause the antigens to clump. So let's say these are all identical antigens. It's one type of particular bacterial organism that's considered foreign to the body. These antibodies are binding to two identical epitopes that are found on two different antigens. Well, the whole point of this is if your immune system cells and the components of the immune system are able to round up these antigens a lot easier, it makes eliminating a whole bunch of these antigens all at one time a lot easier. So let's say there's another cell that comes in for phagocytosis. Instead of having to individually phagocytize every single bacterial cell, right, the antibodies did the job of rounding them all up. And so now this phagocyte can go ahead and eat or take in like a whole bunch of these antigens all at the same time. Here's a different mechanism of action that antibodies are taking. You can see again, it's binding to some foreign bacterial cell. This process here is called opsonization. And you can see that this antigen is coated with different types of antibodies. These are all different antibodies that are binding to different epitopes. And what this does, again, it acts as a signal to the cells of your immune system to come by, say a phagocyte, to come by and recognize that, oh, this foreign object is coated with antibodies, therefore this must be something I need to target for destruction. So the antibodies act as kind of a red flag, and it signals to the cells in your immune system that this is a problem and it needs to be eliminated. We saw that a number of bacterial cells and their associated toxins work initially, and viruses too, work initially by having to bind to some portion on the surface of your cells, right? There has to be a specific recognition molecule that either the virus or the toxic molecule or the bacterial cell latches onto. And so in order for, let's say, a virus to reproduce, this interaction needs to take place in order for the virus to enter the cell to make more of itself. If a virus never enters the cell, it can't reproduce. So if you somehow prevent the virus from binding, then the virus can never reproduce. Same thing with the mechanism of toxins. If the toxin can't enter the cell to disrupt some aspect of cell physiology, then you won't get the associated symptoms associated with that toxin. So the diagram that you're looking at is neutralization of the foreign object through binding by antibodies. So IgGs, which are one class of antibodies, can bind to whatever the antigen is and inactivate those microbes or those toxins by preventing that virus or that bacteria or that toxin from binding to and adhering to host cells. And again, if the virus never attaches to the cell, it never infects the cell. If the toxic molecule never attaches to the cell, it never causes some change in physiology. The diagram that you're looking at on your right is referred to as antibody-dependent cell-mediated cytotoxicity. Antibody-dependent cell-mediated cytotoxicity. So it looks very similar to opsonization, where here you have a large parasitic organism, and you can see that it's covered with antibodies, and the fact that it's covered with antibodies is an indication to your white blood cells that this object, whatever it is, needs to be eliminated. But some of these foreign entities are too big to be phagocytized, right? So bacterial cells are really small, and they can be phagocytized by your eukaryotic cells. But think of like some protozoans, maybe they're really large, or a helminth, which is a multicellular eukaryotic organism. It's a microscopic worm, but some of these worms can be as big as like you know, um, a centimeter in length. So how can one of your itty bitty white blood cells phagocytize like a large helminth like that? Instead, what happens is the large antigen is coated with antibodies, right? So this is the signal. And then a whole bunch of other cells, a variety of other cells in the immune system, recognize that this thing has been coated with antibodies, and then they bind to these antibodies. You can see it here. There's binding of the antibody by macrophage, binding of to the antibody by eosinophil. And instead of engaging in phagocytosis, which obviously this cell is too small to phagocytize this larger thing, um, these other cells in your immune system will begin to release chemical signals, including some enzymes that will cause this target cell to be lysed. So some of these things include lytic enzymes that will be involved in disrupting the membrane of this organism. And if the membrane of this organism is disrupted, then it can eventually lyse. And so there's a lot of enzymes and other chemicals that are released that can do damage to this antigen. All right, T cells. Remember, the adaptive immune response is broken up into humoral immunity, which deals with antibodies, antibody production in B cells, and cell-mediated immunity, which is moderated by your T cells. So humoral antibodies are effective against freely circulating microbes, so things that are occurring outside of your cells. So any viruses that are roaming around freely or bacterial cells that are roaming around freely are going to be taken out by the humoral immune response. But what about any type of microbe, like a virus or an intracellular pathogen that has already gained access into one of your cells? Your immune system doesn't want to destroy your own cells, right? The immune system has to be able to recognize your cells from anything that's not your cells. This is referred to as self versus non-self. So if a virus has infected one of your cells, the surface of your cell is recognized by all the other cells as being self, but clearly a virus infected cell, there's something wrong with it. So there's another aspect of your immune system that will target and eliminate these intracellular pathogens. T cells evolved in response to these intracellular pathogens. They also allow for the immune system to recognize non-self cells like cancerous cells. So a cell that's infected by a virus is going to make more virus particles, and obviously the rest of the body doesn't want that. A bacterial cell that has invaded one of your cells can reproduce or cause damage to that particular cell, and your body doesn't want that to happen either. What if one of your cells isn't infected by a virus and isn't infected by a bacterial cell, but has just become cancerous? So genetically something goes wrong with it, and it begins to reproduce when it's not supposed to reproduce. The body recognizes those cells as well as being abnormal, and will go ahead and attempt to eliminate those abnormal cells. So T cells are specific for certain antigens, right? You'll see in the next couple slides that they are capable of binding to particular antigens. T cells develop from stem cells in the bone marrow, and then they migrate to and eventually mature in this organ called the thymus. You can see that the thymus is here. It's like located right behind the heart. From here, the T cells will migrate to lymphoid tissue, and then this is scattered throughout the mucous membranes that line the gastrointestinal tract, respiratory tract, urinary, and reproductive tract. So these are all, as we saw in a previous chapter, these are all portals of entry that need to be protected. And so this additional system, right, within the adaptive immune response will help protect against microbes, in particular, the intracellular microbes. The body's ability to make new T cells decreases or declines as you get older. So this is why you have the very vulnerable populations are the very elderly, because their immune system doesn't work as well, because their T cells don't work as well. And the younger individuals in the populations, like these kids and these newborns, again, because they haven't developed their adaptive immune response. They haven't encountered these things that will help build up their immunity. The T cells in the thymus become less active as you get older. In fact, the thymus progressively gets smaller as you get older. What we see is that there's fewer B cells that are produced in the bone marrow. And so overall, your immune system weakens or declines as you get older, making you more susceptible to infections and other types of diseases. There are different classes of T cells. And so there are T cells that play a role in humoral immunity. So again, remember, humoral immunity deals with the subset of the adaptive immune response that's involved in production of antibodies. And so primarily that falls under the purview of B cells. But if you remember the diagram that shows you the activation of B cells, B cell activation is assisted by T cells. So there are certain class of T cells that help with B cells in making antibodies. And then there's other types of T cells that are essential for cell-mediated immunity or cellular immunity. And so cell-mediated immunity is not involved in antibody production. Instead, what happens is that these classes of T cells interact directly with the antigen. Again, there is no antibody involved in this. And there's two classes of T cells, right? There's what we call T helper cells or TH cells. And then there's T cytotoxic cells or TC cells. T cells can be differentiated by the molecules that are found embedded on their cell membrane. So these glycoproteins that are embedded on the cell membrane. And so the differentiation is moderated by these groups of proteins called clusters of differentiation. And so the main ones are called CD4 positive T cells. These are T helper cells. So CD4 positive T cells are T helper cells. These types of cells are the ones that are infected by HIV. And so the reason why HIV is an immune disorder is because it weakens the immune system by causing these cells to decrease over a period of time, these CD4 positive T cells. CD4 positive T cells, or these T helper cells, will bind to an MHC class II protein on certain types of cells, MHC class II. The other major type of T cells are the CD8 positive T cells. These are cytotoxic T cells. And CD8 positive T cells bind to an MHC class I protein. So CD4 positive T cells, MHC class II, and CD8 positive T cells bind to MHC class I. At the end of it, at the end of this lecture, you'll see an animation, which hopefully should provide an overview on the entire chapter, and then also a slide at the end of this chapter that summarizes everything that's going on. So here are T helper cells, and the T helper cells, which are these CD4 positive T cells, are interacting with this is one of your cells, right? This is a type of antigen presenting cell, or an APC cell. So the T cell receptor on the CD4 positive T cell will recognize an antigen that has been processed and presented by an MHC class II protein on an antigen presenting cell. So the antigen presenting cell is one of your cells. Let's say the cell has been, this cell has taken in this antigen. right? So now this antigen is brought inside of this cell, and this antigen is then processed. The antigen presenting cell disassembles this antigen, breaks it apart, and by processing the antigen, it will take a fragment of the antigen. You can see here this yellow molecule, or actually these yellow triangles, and these are your MHC class 2 proteins. So the antigen is broken down, it's processed here, and then the MHC class 2 protein will take a portion of that antigen and bring it to the surface of the antigen presenting cell. So this cell presents a fragment of the antigen. This fragment of the antigen is recognized by a T cell receptor on, in this case, a CD4 positive T cell. Because this is a MHC class 2 protein, it's recognized by a CD4 positive T cell. And so this T helper cell then can stimulate the production of antibodies. So it will release chemical signals called cytokines, and this can result in the generation of antibodies. It can signal to other cells to act in phagocytosis. So basically signals to other cells to be on the lookout for this, right, that had whatever antigen that was presented on it. Or it can signal to other cells that play a role in opsonization. So the idea is this is the foreign object. It was brought in by one of your good guy cells, an antigen presenting cell. A portion of it is presented, it's like offered to a T helper cell. The T helper cells then amplify the signal out to all the other cells in the immune system to be on the lookout for this particular thing. Here's how CD8 positive cells work. CD8 positive, oh, sorry, animated out of sequence. These are precursors to cytotoxic T lymphocytes. So you'll see here that cytotoxic T lymphocytes are involved in the direct killing and destruction of an infected cell. So cytotoxic T lymphocytes, or these CD8 positive T cells, will recognize and kill non-self cells, cells that are considered non-self. So maybe the cells can now become cancerous, or it become infected with a virus or something. So it now is acting as non-self. So this cell was infected by a pathogen. The antigen was processed. The antigen is presented on the surface of this cell by an MHC class 1 protein. So see the MHC class 1 protein is here presenting a portion of the antigen. These CD8 positive T cells will recognize the MHC class 1 protein and the antigen, and then will generate and become a cytotoxic T lymphocyte. And as such, as a cytotoxic T lymphocyte, can release chemicals like perforin and granzyme that will cause this cell, the one that had originally been infected by a virus, to now die. And if the cell dies, it can't be used to make more virus particles. So the perforin acts as a molecule that will form pores within the cell, and granzyme is a protease that induces apoptosis. So recall that apoptosis is programmed cell death. And so this original cell that you see here had been infected. It can't be fixed. And so for the good of all the other cells, this cell is going to be scheduled for destruction. Okay, cytokines. Cytokines are chemical signals, and it literally breaks down to cyto refers to cell, kine refers to movement. So these are chemical messengers of the immune cells. And so you've already seen a number of these examples already. There are these molecules that are released by cells that can then tell other cells what to do and how to respond. And so there's different versions, different classes of these cytokines. They all are soluble proteins or glycoproteins, and they're produced in response to some type of stimulus, right? And then they since they are a protein or a glycoprotein that has to be received by the target cell, it's receptor specific. So some other cell has to be able to recognize this particular type of cytokine. And so these are the different versions of the classes of the cytokines. We have interleukins, or ILs. And so you might have seen some examples of interleukins, IL-1, IL-2, for example. These are cytokines that occur between white blood cells. Chemokines are cytokines that induce migration of leukocytes into infection areas. This is important for the inflammation process, and we talked about inflammation in the previous chapter. Interferons are a class of cytokines that are abbreviated as IFNs, and interferons are cytokines that interfere with viral replication. And another class of cytokines called tumor necrosis factors, or TNFs, and this acts as a strong factor in the inflammatory reactions of autoimmune diseases. Okay. Memory. What you're looking at is a graph that shows you how much of that particular antibody occurs within the serum of that patient over a period of time. So at time 0 is when the individual had been exposed to whatever that antigen is. And so of course, it takes some time for the immune system to mount a response. And you can see that, you know, within 7 to 14 days, an immune response is mounted. And the different antibody classes will respond differently. So time 0 is the initial contact with the antigen. You can see here the first week or so, no detectable antibodies for 4 to 7 days. It takes time for the immune system to work and to trigger the production of antibodies. And so you get a slow rise of the antibody titer. Titer refers to the concentration. So in this case, it's the concentration of the antibodies that's in the serum. Initially, you get an increase in the rise of these IgM, the class of immunoglobulins called IgMs, and then IgGs. So you can see here within the first 3 weeks, IgM peaks, right? And then IgG is the class of antibodies that will respond secondarily. The secondary response to the same antigen is based upon immunologic memory. So your immune system here has fought off the antigen and won. Let's say you get infected with the same antigen again later on. Upon the second exposure to that same antigen, your immune response occurs a lot faster, and as you can see from the graph, it's more intense. So the antibody-mediated immune response is intensified. You can see a massive spike in the IgGs, and you get a faster, longer, more intense response from your immune system the second time around upon previous exposure. So this is what the basis for vaccinations is so that at an early age, you can be exposed to something in a weak version of whatever that antigen is. And so your body can build up antibodies and develop memory against that thing that you've been injected with in vaccinations, so that if you ever encountered that antigen for real later on in life, you already have immunologic memory against it, so your immune system can respond a lot faster. So there's different types of adaptive immunity. There's what we call actively acquired adaptive immunity. And actively acquired adaptive immunity is where you get exposed to a foreign substance, and this results in the stimulation of an immune response, right? So you live your life, you encounter something naturally or actively, and you develop an immune response because of something that you had encountered. So let's say that, you know, you lived your life and you had never been vaccinated against chickenpox, and then you got the virus that causes chickenpox. Then you got chickenpox, now you developed immunity against chickenpox in the future. That's actively acquired adaptive immunity. Passively acquired adaptive immunity refers to a transfer of antibodies from one person to another. So here, you didn't actively acquire the infection. You got the antibodies from somebody else that had developed the antibodies against the infection. So this is what's happening, or this is what is attempted to be tried out with individuals that are had already been infected with coronavirus, and had recovered. And so the thinking goes that individuals that had recovered from coronavirus complications, they should have immunologic memory against it. So in their serum, they should have antibodies against it. And so maybe those antibodies can be transferred passively to an individual that is now suffering from coronavirus complications. And so instead of waiting for the antibodies to develop in the new patient, the antibodies are coming from a patient that had already recovered from that previous illness. And so it allows for the recipient patient to also recover more quickly. So the problem with this is, since you aren't making the antibodies because you didn't actively acquire that infection, those antibodies that were transferred to you through a transfusion will only last for a certain period of time, right? So a number of weeks. Naturally acquired immunity, there are two different versions. You can get active naturally acquired immunity. This is where, again, you get exposed to the antigen. You live your life, you naturally are exposed to it, and then you develop immunity against it. You get sick, and then you recover, and now your body has antibodies against it. Passive naturally acquired immunity results from a transfer of antibodies from one individual to another. So let's say that natural passive acquired immunity occurs from mother to infant, so upon breast milk or transplacentally. So you have the mother that had developed an immune response to whatever over the course of living her life is passing on the antibodies from her breast milk to the newborn infant. And so again, remember, this newborn hasn't encountered anything yet because it hasn't lived that long. And so it's receiving passively acquired immunity through breast milk or transplacentally. Again, this only lasts as long as the antibodies are willing to exist within the system. Remember, the B cells in this individual, the recipient, hadn't gone into memory yet because those B cells were never activated in the first place. Artificially acquired immunity can be active. And this is where vaccinations come in. So you can be introduced to an antigen, and vaccinations will do that artificially. So when you go to the doctors and get a vaccination for something, they're putting a foreign molecule inside of you, whether it's a portion of the bacteria or a portion of the toxin, and it's allowing for your immune system to develop antibodies against what was artificially put inside of you. So the antigens are killed off, or excuse me, the antigens that are introduced are a weakened form or inactivated or killed off version of the thing that you would encounter naturally. So vaccinations typically are like softball pitches, right? They introduce some type of molecule that will allow for your immune system to very easily mount an immune response against, where you won't get sick. But if you encounter that real antigen later on in life, you already have immunologic memory against it. You can also get passive artificially acquired immunity, and this is from an injection of antibodies, not antigens, into the body. So remember, this is artificial, right? So taking antibodies from an individual that recovered from one illness and taking the serum from that individual that has now the antibodies and injecting it into another individual, that is artificial, right? That's not natural. That's natural would be like from mother to child, and that's a natural process. So an injection of antibodies, not antigens, into the body from an organism that is already immune to that particular disease. So here's the summary slide. This is the end of chapter 17 now, or the second part of chapter 17. And this is an overview showing you the two parts of adaptive immunity. So on the left in blue is the humoral immune response, which again deals exclusively with antibody production. So there are B cells and there are T cells that can come in and help with the production of antibodies and memory B cells. And then the other part of adaptive immunity is cell-mediated immunity. And you can see here, there's no involvement of antibodies here. These are T cells that play a role in the direct destruction of whatever this infected cell is here. Okay, so I will publish the video. Make sure that you take a look at it. The video should be an overview of all of the parts of the immune response system. And so make sure you take a look at it. Take your notes, and if you have any questions, just send me an email. Otherwise, this ends the recording for chapter 17, Adaptive Immunity. I'll see you next time.